In this module, we're going to be discussing enzymes. And so enzymes control and mediate a lot of the chemistry within the cell, basically most of the chemistry within the cell, okay, and the chemical transformations within. So enzymes are catalysts, okay, and they're protein-based catalysts. So we've been discussing proteins already in the last module, but they also perform chemistry, and what they do, what the definition of a catalyst is, it increases the rate of a reaction and does not get consumed. Okay, so for instance, on the left-hand side, we have an enzyme that's called catalase. This is a very efficient enzyme that catalyzes the what's called the disproportionation. So it's like two redox reactions in one. So it takes hydrogen peroxide as what's known as a substrate. So when we talk about enzymes, we talk about substrates instead of ligands, okay? And it converts the substrate to two products. One is one of the peroxides gets oxidized to dioxygen, and the other get converted to water, okay? Two molecules of water. And so this catalyzes this reaction, and as you notice, the catalase itself does not show up in either the reactant or the product side. Okay, it's not consumed. It is performing the chemistry and increasing the rate of this reaction compared to the uncatalyzed reaction, but it's not. For instance, it's not. You're not going to be considered either a reactant or a product. It does this by doing cycles. Okay, so catalysts and enzymes, since enzymes are just protein catalysts, does this by going through cycles. So we have what's called the resting state, and in the resting state, this is an iron 3 form. Right, this reacts with peroxide. It forms what's known as an intermediate. Okay, this is an intermediate state that's important for the reaction. This intermediate state reacts with another molecule of hydrogen peroxide to form oxygen and water, and then it returns back to the resting state. And as long as there's hydrogen peroxide around, this will continue to go in this cycle until no hydrogen peroxide is available, and then it'll return back to the resting state, which is this iron 3 form. Okay, so it just cycles, and it regenerates itself and closes the cycle and regenerates itself again, and therefore never gets consumed within the reaction. So there are different types of enzymes, and so these are the major classes of enzymes. So the first one is an oxidoreductase. So one example of an oxidoreductase is alcohol dehydrogenase that catalyzes the following reaction of ethanol to acetaldehyde. And we've seen this before, and this is an oxidation of ethanol to acetaldehyde, and there's a corresponding reduction of NAD plus to NADH. Okay, so you can classify these by noting that they're redox reactions. So if you look at the oxidation states for these, for the carbons and the ethanol, the carbon on the left-hand side, the far left-hand side of ethanol is -3, the middle carbon is -1, and then on the right-hand side, the acetaldehyde, if we look at the corresponding carbons, this carbon stays at an oxidation state of -3, and the middle and the second one turns to +1. So there's a oxidation state change from -1 to +1. So it's a two electron oxidation at that carbon, which corresponds with the 2 electron reduction of NAD plus to NADH. So this is a redox reaction, and so oxidoreductases are the enzymes that catalyze redox reactions. All right. Transferases are a second major class, and so these participate in functional group transfer. All right, and this is an example. This is hexokinase. So a functional group from ATP, which can donate phosphate, is added to glucose, which is the substrate is a substrate of the reaction of the hexokinase, and the phosphate group from ATP gets added, and so that's shown here in pink. And so it's just transferring a phosphate group from ATP to D-glucose to form D-glucose 6-phosphate. Third major class is a hydrolase. Okay, as the name implies, this involves water. And in fact, what this does is it adds water across a bond to cleave the bond. Okay, so one example of this is carboxypeptidase A, which participates in peptide bond cleavage. So if we have the peptide bond here, and so this is a C terminus of a polypeptide, okay, where the C terminal amino acid is shown here in pink, and so this is the last peptide bond of the polypeptide chain. Okay, so there's our CO and our NH. Water is added across the peptide bond to release this amino acid, the C-terminal residue, so that gets regenerated to whatever the amino acid is. So let's say this was a glycine residue. This is now cleaved to form glycine, and then now the polypeptide has one less amino acid on its chain. Okay, so the key here is that this is a hydrolysis reaction. So this is addition of water across the bond to cleave it, and that's indicated in the name of hydrolase. Okay, fourth class of enzyme are the Lyases. Okay, so these cleave bonds as well, but they're not hydrolysis reactions. They're also not redox reactions, even though they kind of look like them. So as an example, this is pyruvate decarboxylase. So pyruvate is shown here as the substrate, and then the product is carbon dioxide and acetaldehyde. So you have a loss of carbon dioxide, or the carboxylate group gets converted to carbon dioxide, and so that kind of looks like a redox reaction. But if you look at the sum total of at least these two carbons here, you can do this for all the carbons, you'll still get a zero net change in oxidation state. But you can look at the carbons, these two carbons on the pyruvate, which are +3 and +2 for the first two carbons on the left. And then what you see is those convert to the carbon dioxide, which is cleaved, now has an oxidation state of +4, and this second carbon has an oxidation of +1. So the net oxidation state is still +5 on the left-hand side, +5 on the right-hand side. Okay, and so that's not actually a redox reaction, but it kind of looks like one if you don't add up all of the oxidation states for all those. So those cleave the bonds. So this cleaves bonds or adds across double bonds for Lyases. Okay. Isomerases, as the name suggests, Isomerases will change isomers of a molecule. So the example here is malleate isomerase. So it's a cis-trans isomerization. Okay. So for malleate, we have the carboxylate groups are cis across the double bond, and using the malleate isomerase, this takes malleate as the substrate and converts it to the product fumarate. And so the only difference here is now the carboxylate groups are trans to each other across the double bond. Okay, so this is one example of isomerase where there's a conversion between the cis-trans isomer, but other isomers can also other isomerizations can also be done, for instance, stereoisomer or other configurational isomers. And finally, ligases, such as pyruvate carboxylase, join groups. Okay, this is also not a redox reaction, so you'll see carbon dioxide and a carboxylate, which technically would change the oxidation state of that carbon, but the overall net oxidation states there are not are a net zero change, much in the same way as Lyases. And in this case, this is joining groups. Okay, so for instance, carbon dioxide. So this is a carboxylation now that's going to react with the substrate pyruvate. ATP is involved here, okay, as a cofactor, and then, or as a co-substrate, and then this is the product of that is oxaloacetate. Okay, and so those are the major activities or major classes of enzymes.